Toll-like receptors are a class of proteins that play a key role in the innate immune system. They are single, membrane-spanning, non-catalytic receptors usually expressed in sentinel cells such as macrophages and dendritic cells, that recognize structurally conserved molecules derived from microbes. Once these microbes have breached physical barriers such as the skin or intestinal tract mucosa, they are recognized by TLRs, which activate immune cell responses. The TLRs include TLR1, TLR2, TLR3, TLR4, TLR5, TLR6, TLR7, TLR8, TLR9, TLR10, TLR11, TLR12, and TLR13. They receive their name from their similarity to the protein coded by the Toll gene identified in Drosophila in 1985 by Christiane Nüsslein-Volhard. The researchers were so surprised that they spontaneously shouted out in German, "Das ist ja toll," which translates as "that's great." Diversity. TLRs are a type of pattern recognition receptor and recognize molecules that are broadly shared by pathogens but distinguishable from host molecules, collectively referred to as pathogen-associated molecular patterns. TLRs together with the interleukin-1 receptors form a receptor superfamily, known as the interleukin-1 receptor/toll-like receptor superfamily. All members of this family have in common a so-called TLR domain. 3 subgroups of TLR domains exist. Proteins with subgroup 1 TLR domains are receptors for interleukins that are produced by macrophages, monocytes, and dendritic cells and all have extracellular immunoglobulin domains. Proteins with subgroup 2 TLR domains are classical TLRs, and bind directly or indirectly to molecules of microbial origin. A third subgroup of proteins containing TLR domains consists of adapter proteins that are exclusively cytosolic and mediate signaling from proteins of subgroups 1 and 2. TLRs are present in vertebrates, as well as in invertebrates. Molecular building blocks of the TLRs are represented in bacteria and in plants, and plant pattern recognition receptors are well known to be required for host defense against infection. The TLRs thus appear to be one of the most ancient, conserved components of the immune system. In recent years TLRs were identified also in the mammalian nervous system. Members of the TLR family were detected on glia, neurons and on neural progenitor cells in which they regulate sulfate decision. Discovery. When microbes were first recognized as the cause of infectious diseases, it was immediately clear that multicellular organisms must be capable of recognizing them when infected and, hence, capable of recognizing molecules unique to microbes. A large body of literature, spanning most of the last century, attests to the search for the key molecules and their receptors. More than 100 years ago, Richard Pfeiffer, a student of Robert Koch, coined the term endotoxin to describe a substance produced by gram-negative bacteria that could provoke fever and shock in experimental animals. In the decades that followed, endotoxin was chemically characterized and identified as a lipopolysaccharide produced by most gram-negative bacteria. This lipopolysaccharide is an integral part of the gram-negative membrane and is released upon destruction of the bacterium. Other molecules were shown in turn to provoke host responses that are normally protective. However, these responses can be detrimental if they are excessively prolonged or intense. It followed logically that there must be receptors for such molecules, capable of alerting the host of the presence of infection, but these remained elusive for many years. Toll-like receptors are now counted among the key molecules that alert the immune system to the presence of microbial infections. They are named for their similarity to toll, a receptor first identified in the fruit fly Drosophila melanogaster, and originally known for its developmental function in that organism. In 1996, toll was found by Jules A. Hoffman and his colleagues to have an essential role in the fly's immunity to fungal infection, which it achieved by activating the synthesis of antimicrobial peptides. The plant homologs were discovered by Pamela Ronald in 1995 and Thomas Bowler in 2000. The first reported human toll-like receptor was described by Nomura and colleagues in 1994, mapped to a chromosome by Taguchi and colleagues in 1996. Because the immune function of TLR and Drosophila was not then known, it was assumed that TLR might participate in mammalian development. However, in 1991 it was observed that a molecule with a clear role in immune function in mammals, the interleukin-1 receptor, also had homology to Drosophila TLR. The cytoplasmic portions of both molecules were similar. In 1997, Charles Janeway and Roland Medzhitov showed that a TLR-like receptor now known as TLR4 could, when artificially ligated using antibodies, induce the activation of certain genes necessary for initiating an adaptive immune response. TLR4 function as an LPS sensing receptor was discovered by Bruce A. Bihler and colleagues. These workers used positional cloning to prove that mice that could not respond to LPS had mutations that abolished the function of TLR4. This identified TLR4 as one of the key components of the receptor for LPS. In turn, the other TLR genes were ablated in mice by gene targeting, largely in the laboratory of Shizuo Akira and colleagues. Each TLR is now believed to detect a discrete collection of molecules some of microbial origin, and some products of cell damage and to signal the presence of infections. On October 3, 2011, Dr. Bihler and Dr. Hoffman were awarded the Nobel Prize in Medicine or Physiology for their work. DRS. Hoffman and Akira received the Canada Gairdner International Award in 2011. Extended family. It has been estimated that most mammalian species have between 10 and 15 types of TLR-like receptors. 13 TLRs have been identified in humans and mice together, and equivalent forms of many of these have been found in other mammalian species. However, equivalents of certain TLR found in humans are not present in all mammals. For example, a gene coding for a protein analogous to TLR10 in humans is present in mice, but appears to have been damaged at some point in the past by a retrovirus. On the other hand, mice express TLRs 11, 12, and 13, none of which is represented in humans. Other mammals may express TLRs that are not found in humans. Other non-mammalian species may have TLRs distinct from mammals, as demonstrated by TLR14, which is found in the Takaifugu pufferfish. This may complicate the process of using experimental animals as models of human innate immunity. TLRs in Drosophila immunity. Drosophila melanogaster has only innate immune responses. Response to fungal or bacterial infection occurs through two distinct signaling cascades, one of which is TLR pathway and the other is immune deficiency pathway. The TLR pathway is similar to mammalian TLR signaling, but unlike mammalian TLRs, TLR is not activated directly by PAMPs. Its receptor ectodomain recognizes cleaved form of the cytokine SPA currency TZLE, which is secreted in the hemolymph as inactive dimeric precursor. TLR receptor shares the cytoplasmatic TIR domain with mammalian TLRs, but ectodomain and intracytoplasmatic TLR are different. This difference might reflect a function of these receptors as cytokine receptors rather than PRRs. TLR pathway is activated by different stimuli, such as gram-positive bacteria, fungi and virulence factors. First, the SPA currency TZLE processing enzyme is activated in response to infection and cleaves SPA currency TZLE. Cleaved SPA currency TZLE then binds to TLR receptor and cross-links its ectodomains. This triggers conformational changes in receptor resulting in signaling through TLR. Signaling now is very similar to mammalian signaling through TLRs. TLR-induced signaling complex is formed, comprising MyD88, tube and PEL, the ortholog of mammalian IRAK. Signal from ticks is then transduced to cactus. Phosphorylated cactus is polyubiquitilated and degraded, allowing nuclear translocation of DIF and induction of transcription of genes for antimicrobial peptides such as drosomycin. TLR2 has also been designated as CD282. TLR3 is the only TLR-like receptor which does not use the MyD88-dependent pathway. Its ligand is retroviral double-stranded RNA, which activates the TRIF-dependent signaling pathway. To explore the role of this pathway in retroviral reprogramming, knockdown techniques of TLR3 or TRIF were prepared, and results showed that only the TLR3 pathway is required for full induction of target gene expression by the retrovirus expression vector. This retroviral expression of 4 transcriptional factors induces pluripotency in somatic cells. This is supported by study, which shows that efficiency and amount of human iPSC generation, using retroviral vectors, is reduced by knockdown of the pathway with peptide inhibitors or shRNA knockdown of TLR3 or its adapter protein TRIF. Taken together, stimulation of TLR3 causes great changes in chromatin remodeling and nuclear reprogramming, and activation of inflammatory pathways is required for these changes, induction of pluripotency genes and generation of human-induced pluripotent stem cells colonies. TLR11, as mentioned above, human cells do not express TLR11, but mice do. It has been characterized that mouse-specific TLR11 recognizes aeropathogenic E. coli and apiconfloxan parasite Toxoplasma gondii. ETA-1 trademark S ligand is profilin protein in case of Toxoplasma, but ligand for E. coli is still not known. Recently there was found another enteropathogen which ligand is bound by TLR11. It is Salmonella spp. This gram-negative flagellated bacteria causes food and waterborne gastroenteritis and typhoid fever in humans. TLR11 in mouse intestine recognizes this flagellin which causes dimerization of the receptor, activation of NFIB and production of inflammatory cytokines. TLR11 deficient mice are efficiently infected with orally administered Salmonella typhi. S. typhi does not normally infect mice. It is human obligatory pathogen that causes typhoid fever, which affects more than 20 million people and causes more than 220,000 deaths per year. Because of this, studies were carried out and it was found that TLR/mice can be immunized against S. typhi and they are used as an animal model for studying immune responses against this pathogen and for the development of vaccines that could be possibly used in the future. Ligands. Because of the specificity of toll-like receptors they cannot easily be changed in the course of evolution, these receptors recognize molecules that are constantly associated with threats and are highly specific to these threats. Pathogen-associated molecules that meet this requirement are thought to be critical to the pathogen's function and difficult to change through mutation. They are said to be evolutionarily conserved. Somewhat conserved features in pathogens include bacterial cell surface lipopolysaccharides, lipoproteins, lipopeptides, and liarabinomanan. Proteins such as flagellin from bacterial flagella, double-stranded RNA of viruses, or the unmethylated CpG islands of bacterial and viral DNA, and also of the CpG islands found in the promoters of eukaryotic DNA, as well as certain other RNA and DNA molecules. For most of the TLRs, ligand recognition specificity has now been established by gene targeting, a technique by which individual genes may be selectively deleted in mice. See the table below for a summary of known TLR ligands. Endogenous ligands. The stereotypic inflammatory response provoked by toll-like receptor activation has prompted speculation that endogenous activators of toll-like receptors might participate in autoimmune diseases. TLRs have been suspected of binding to host molecules including fibrinogen, heat shock proteins, HMGB1, extracellular matrix components and self-DNA. These endogenous ligands are usually produced as a result of non-physiological cell death. Signaling. TLRs are believed to function as dimers. Though most TLRs appear to function as homodimers, TLR2 forms heterodimers with TLR1 or TLR6, each dimer having a different ligand specificity. TLRs may also depend on other co-receptors for full ligand sensitivity, such as in the case of TLR4's recognition of LPS, which requires MD2. CD14 and LPS binding protein are known to facilitate the presentation of LPS to MD2. A set of endosomal TLRs comprising TLR3, TLR7, TLR8 and TLR9 recognize nucleic acid derived from viruses as well as endogenous nucleic acids in context of pathogenic events. Activation of these receptors leads to production of inflammatory cytokines as well as type I interferons to help fighting viral infection. The adapter proteins and kinases that mediate TLR signaling have also been targeted. In addition, random germline mutagenesis with ENU has been used to decipher the TLR signaling pathways. When activated, TLRs recruit adapter molecules within the cytoplasm of cells in order to propagate a signal. 4 adapter molecules are known to be involved in signaling. These proteins are known as MyD88, TRAP, TRIF, and TRAM. TLR signaling is divided into 2 distinct signaling pathways, the MyD88-dependent and TRIF-dependent pathway. MyD88-dependent pathway. The MyD88-dependent response occurs on dimerization of the TLR receptor, and is utilized by every TLR except TLR3. Its primary effect is activation of NFIB and mitogen-activated protein kinase. Ligand binding and conformational change that occurs in the receptor recruits the adapter protein MyD88, a member of the TLR family. MyD88 then recruits IRAK4, IRAK1 and IRAK2. IRAK kinases then phosphorylate and activate the protein TRAF6, which in turn polyubicinates the protein TAK1, as well as itself in order to facilitate binding to IKKI². On binding, TAK1 phosphorylates IKKI², which then phosphorylates EB, causing its degradation and allowing NFIB to diffuse into the cell nucleus and activate transcription and consequent induction of inflammatory cytokines. TRIF-dependent pathway. Both TLR3 and TLR4 utilize the TRIF-dependent pathway, which is triggered by dsRNA and LPS, respectively. For TLR3, dsRNA leads to activation of the receptor, recruiting the adapter TRIF. TRIF activates the kinases TBK1 and IPK1, which creates a branch in the signaling pathway. The TRIF-TBK1 signaling complex phosphorylates IRF3, allowing its translocation into the nucleus and production of interferon type I. Meanwhile, activation of IPK1 causes the polyubiquitination and activation of TAK1 and NFIB transcription in the same manner as the MyD88-dependent pathway. TLR signaling ultimately leads to the induction or suppression of genes that orchestrate the inflammatory response. In all, thousands of genes are activated by TLR signaling, and collectively, the TLRs constitute one of the most pleiotropic yet tightly regulated gateways for gene modulation. TLR4 is the only TLR that uses all 4 adapters. Complex consisting of TLR4, MD2 and LPS recruits TIR domain-containing adapters TIRAP and MyD88 and thus initiates activation of NFIB and MAPK. TLR4 MD2 LPS complex then undergoes endocytosis, and in endosome it forms a signaling complex with TRAM and TRIF adapters. This TRIF-dependent pathway again leads to IRF3 activation and production of type I interferons, but it also activates late-phase NFIB activation. Both late and early-phase activation of NFIB is required for production of inflammatory cytokines. Summary of known mammalian TLRs. Toll-like receptors bind and become activated by different ligands, which, in turn, are located on different types of organisms or structures. They also have different adapters to respond to activation and are located sometimes at the cell surface and sometimes to internal cell compartments. Furthermore, they are expressed by different types of leukocytes or other cell types. Activation and effects. Following activation by ligands of microbial origin, several reactions are possible. Immune cells can produce signaling factors called cytokines, which trigger inflammation. In the case of a bacterial factor, the pathogen might be phagocytosed and digested, and its antigens presented to CD4+ T cells. In the case of a viral factor, the infected cell may shut off its protein synthesis and may undergo programmed cell death. Immune cells that have detected a virus may also release antiviral factors such as interferons. The discovery of the toll-like receptors finally identified the innate immune receptors that are responsible for many of the innate immune functions that had been studied for many years. It is interesting to note that TLRs seem to be involved only in the cytokine production and cellular activation in response to microbes, and do not play a significant role in the adhesion and phagocytosis of microorganisms. Schmidt et al. Demonstrated that TLR4 is involved in the development of contact allergy to nickel in humans. By binding to two non-conserved histidines, H456 and H458, Me2+ cross-links the two receptor monomers, TLR4, and MD2, triggering formation of a dimer that structurally resembles the one induced by lipopolysaccharide. That, in turn, activates the pro-inflammatory intracellular signal transduction cascades. Said et al. Showed that TLR ligands cause an IL-10-dependent inhibition of CD4 T cell expansion and function by upregulating PD-1 levels on monocytes, which leads to IL-10 production by monocytes after binding of PD-1 by PDL. Toll-like receptors have also been shown to be an important link between innate and adaptive immunity through their presence in dendritic cells. The TLRs 3 and 4 are present on the surface of monocyte-derived dendritic cells and use the MyD88-dependent pathway to produce interleukins 12 and 18, which signal naive T cells to mature into type I helper T cells. These TLRs also use the TRIF pathway to upregulate costimulatory proteins which aid in the differentiation of T cells. TLRs 7 and 9 are present on the endosome of plasmacytoid dendritic cells. These proteins solely make use of the MyD88-dependent pathway to produce interleukins for the maturation of naive T cells to type I helper T cells. Evelyn A. Kurt-Jones et al. Also demonstrated TLR4's role in the innate immune response to the respiratory syncytial virus. Cytokine production increased upon exposing human monocytes to RSV. After knocking out CD14, TLR4's coactivator, this response was significantly diminished, as were the responses in C3HEJ mice, a mouse strain with reduced immune responses, and mice deficient in TLR4. Drugs interactions, imiquimod, and its accessory sequimod, are ligands for TLR7 and TLR8. The lipid A-analogon erythrin acts as a TLR4 antagonist. As of December 2009, it is being developed as a drug against severe sepsis. Notes and references. See also, NOD-like receptor, external links, Toll-like receptors at the U.S. National Library of Medicine. Medical subject headings, Toll-ML, Toll-like receptors and ligands database at University of Munich, the Toll-like receptor family of innate immune receptors, Toll-like receptor pathway.